This is the Group Advanced Concepts Presentation 3 by myself, Mariella Roper. This presentation will discuss the properties of hemoglobin and the significance of the heme in hemoglobin. Hemoglobin is a complex protein made up of 4 subunits, more specifically 2 alpha and 2 beta subunits. This structure functions as a pair of alpha-beta dimer, giving hemoglobin its complex quaternary structure. Part of each hemoglobin molecule is 4 heme groups, which are organic molecules that each surround a globin group. Each heme molecule contains a central iron atom that forms 4 bonds to the nitrogen atoms in the chiral rings with the capacity to form 2 additional bonds. These 2 additional bonds are on each side of the heme plane, called the 5th and 6th coordination sites. In hemoglobin, the 5th coordination site is occupied by histidine residues in the protein. However, the 6th coordination site remains unoccupied, only available for binding to oxygen. When oxygen binds to the 6th coordination site in a heme group, the molecule's electrons are substantially rearranged and the heme group becomes planar as opposed to the dome shape of deoxygenated heme groups. As the oxygenated heme groups becomes polar, the 5th coordination group site moves with it and results in a substantial change in quaternary structure. The alpha-beta dimers of hemoglobin rotate approximately 15 degrees with respect to one another and coordinates the transition from the T state to R state for the remaining subunits in the protein, thus effectively raising the hemoglobin's affinity for oxygen. The binding capacity of oxygen to heme varies in different areas of the body and can be directly influenced by allosteric regulators. In the lungs, for example, hemoglobin becomes nearly saturated with oxygen with 98% of binding sites occupied. This is due in part to the lack of 2,3-biphosphoglycerate, an allosteric regulator molecule that, when bonded to hemoglobin, reduces its affinity for oxygen. So in an environment without this regulator, such as in the lungs, hemoglobin's affinity for oxygen is very high and thus can lead to high saturation rates. In the capillaries, however, high oxygen affinity would be unfavorable as oxygen would need to be released. Thus, 2,3-biphosphoglycerate plays an important role as it decreases the binding affinity of oxygen. It actually reduces hemoglobin's oxygen affinity so drastically, in fact, that 66% of oxygen is released instead of the mere 8% when the regulator is not present. Additionally, in the capillaries, hydrogen and carbon dioxide molecules also act as heterotropic effectors that enhance the release of oxygen. This is called the Bohr effect, and in the capillaries is highly favorable since the release of more oxygen means that more oxygen that can be readily diffused into the tissues. The Bohr effect additionally describes the influence of pH and oxygen affinity. Carbon dioxide in the tissues diffuses into red blood cells in order to be carried back to the lungs as waste. However, inside the red blood cell, carbon dioxide reacts with water and forms carbonic acid, which then dissociates and results in a drop in pH in the red blood cell. The drop in pH further reduces the heme group affinity for oxygen, resulting in 77% of the oxygen being released. This is due to the ionic bonds that form due to the lowered pH, particularly because of the protonation of amino acid residues, thus favoring the deoxygenated structure and favoring the release of more oxygen. Hemoglobin adaptations in physiological conditions provides great examples of how form follows function, especially in the influence of other factors such as carbon dioxide concentrations and the presence of regulator molecules. By controlling intake and release of oxygen, the body further maximizes its efficiency and reduces waste in metabolic pathways.